friends and colleagues, ladies and gentlemen, thank you very much for your introduction, Hans. My title is a little bizarre, I know. I'm trying to compare grammar with immunology. Grammar is a science that is more than 2,000 years old, whereas immunology has become a respectable part of biology only during the past 100 years. Though both sciences still face exasperating problems, this lecture attempts to establish an analogy between linguistics and immunology, between the descriptions of language and the description of the immune system. Let me first recall some of the essential elements of the immune system with which I shall be concerned. In 1890, Behring and Kitasato were the first to discover antibody molecules in the blood serum of immunized animals and to demonstrate that these antibodies could neutralize diphtheria toxin and tetanus toxin. They also demonstrated the specificity of antibodies. Tetanus antitoxin cannot neutralize diphtheria toxin, and vice versa. During the first 30 years or more after this discovery, most immunologists believed that all cells in the body of an animal are capable of producing antibodies. And it took, strange enough, until the 1950s before it became clear, and until 1960 before it was demonstrated, that only white blood cells called lymphocytes can produce antibodies. The total number of lymphocytes in the body, in our body and in the body of animals, represent a little more than 1% of the body weight. Thus, it would not be wrong to say that our immune system, I write it on the blackboard, the human immune system equals 10 to the 12 lymphocytes. And if you consider a mouse, which is about 3,000 times smaller than we are, mouse, immune system is the mouse has about 3,000 times fewer lymphocytes, about 3 times 10 to the 8 lymphocytes. This is a very brief description of the system, and it disregards the fact that these lymphocytes interact with other cells of the body. But in my definition, the other cells do not belong to the immune system sensu stricto. And I'd like to draw your attention to the fact that this number of lymphocytes is at least 1 order of magnitude larger than the number of neurons in the nervous system. Also, we should note that the immune system is an organ, but not a nice little round organ like the liver and heart and so. The lymphocytes, they travel among most other cells of our body. They circulate in blood and in lymph, and they occur in larger concentrations in lymph nodes, spleen, thymus, bone marrow, appendix, and so forth. Strangely enough, they seem to be excluded from the brain. The 1960s was a very fruitful decade for immunology. I'll name a few of the discoveries of that decade. In the beginning of the 1960s, the primary structure of antibody molecules was clarified by Porter and Edelman. Then followed the demonstration or the dictum of Burnett, namely that all antibodies made by one single lymphocyte are identical. And finally, towards the end of the decade, it was shown that lymphocytes fall into two classes called T cells and B cells. T lymphocytes and B lymphocytes. And there's about an equal number of each of these. But only the B lymphocytes produce and secrete antibody molecules. I was trying to show you this on a slide, please. So here I have repeated that the human immune system is 10 to the 12 lymphocytes. There are T cells and B cells. The top one of these cells we call resting lymphocytes, and if the resting lymphocyte is triggered or stimulated and gets the right growth factors, maturation factors, and so forth, it will develop into a clone. The B cells carry on the surface receptors, which we now know are antibody molecules, which are stuck into the membrane of the cell. There's about 100,000 of those receptors on each cell. The cells will proliferate, and finally, after a clone size of a certain size has been reached, the cell will secrete about 2,000 antibody molecules per second, and these antibody molecules will be identical or similar to the receptor on the stem cell from which the other cells have arisen. As for the T cells, we also know that they carry receptors that can recognize foreign molecules, antigens. And unfortunately, these receptors are not so well known. The structure of them is not yet known, but in the last few years, great advances have been made towards that. And what is perhaps important to say is that the T cells do not secrete receptor molecules. They don't make antibodies. All antibodies we have in our blood and in our system are made by B cells. The T cells, again, fall into two groups, at least two groups, which are called T helper cells and T killer cells. The T helper cells are mostly, in most situations, are needed to stimulate a B cell to proliferate and to make antibodies. That's why they're called helper cells. And if the helper cells are missing, the B cell cannot be stimulated. The T killer cells are cells which can kill other cells, for instance, cells they don't like virus-infected cells. And they can also prevent, as suppressor cells, the B cells from being stimulated. Of course, when an animal responds to an antigen, it's not only one cell, as they're at the top of the B cells that is stimulated, but many, usually several hundreds of different ones, so that the immune response to an antigen in an animal is characterized by a polyclonal production of antibody molecules. Before looking at the grammar, we can take the slide off, please. I must briefly describe the structure of an antibody molecule. No matter what you try to investigate in biology, it turns out to become increasingly complex. And that's also true for the structure of antibody molecules. The basic element of an antibody molecule, I'll show on the next slide, please, which is quite surrealistic, perhaps. We know it's Y-shaped, and it has a molecular weight of about 150,000 Daltons. This is, of course, a three-dimensional structure, like all molecules and cells that biology has to deal with. And three-dimensionality tends to perplex our mind. Our mind is much more at ease with one-dimensional linear sequences. And this is just sort of a two-dimensional sketch. We can make some important cuts through this molecule. The vertical one divides the molecule into two identical halves. It's symmetrical. The two square cuts separate a constant region in the carboxyl end of the molecule and a variable region, two variable regions that are identical, so that in this respect the molecule is divalent. The constant part, by constant we mean that two different antibody molecules, let's say tetanus antitoxin and diphtheria antitoxin, they may have the same constant part, but they differ in the variable part. So that it's the variable part that determines the specificity of the antibody molecule. Now, the one-dimensional structure of this molecule is well known. Maybe we can have the next slide. The molecule consists of the two symmetrical halves, and in each of these there's a heavy chain, that's a long one, and a light chain, which is a short one, and the light chain has about 214, I think, amino acid residues, and the heavy chain has somewhat more than 400 amino acid residues. It turned out then that the differences between two antibody molecules of different specificity lies in the amino acid sequence of these 100 amino acid residues in the amino end of the light chain and 100 amino acids in the amino end of the heavy chain. So there are about 200 amino acid residues that make up the variable part, and it turned out that the variability of the amino acid sequences is enormous in that region. This insight came immediately when these chemical problems were solved, but they didn't solve our problems. It's like saying that the great variety of words and sentences in a language results from an enormous number of varieties with respect to the sequence of letters or phonemes. Traditionally, the antibody combining site was conceived as a cleft at the end of the variable region somewhere, which recognizes a protuberance in the molecule for which it is specific. And traditionally, all antibody molecules or antibodies were named after the antigen which they can recognize. So anti-diphtheria toxin, or diphtheria antitoxin, anti-sheep red blood cells, anti-TMP, and so forth. I shall try to give you an impression of the size of this system of antigens and specific antibody molecules. Let's first consider macromolecules, let's say of molecular weights exceeding 10,000 Daltons. Maybe polysaccharides, maybe proteins, lipoproteins, nucleic acids, viruses, bacteria. Any molecule you care to mention, any particle, is an antigen that is capable of inducing the immune system to make specific antibodies. Moreover, molecules like nitrophenol, arsenate, in fact, any inorganic molecule you care to mention, if you attach it to a protein, for instance, it will induce the production by our immune system of antibodies that are specific for these inorganic, recognizing these inorganic molecules. And this went so far that even if you synthesize molecules in a laboratory that have never existed in the world, the immune system will make specific antibodies against them. How is this possible? Of course, a mouse has only 3 times 10 to the 8 lymphocytes, let's say 10 to the 8 B cells. So and since every B cell makes only antibodies of one specificity, the repertoire in a mouse cannot be at any given moment more than 10 to the 8 different specificities, and probably less. And we must then realize, since the number of antigens is much larger, that recognition of a foreign molecule is more or less precise and not perfect. And that the same combining site may recognize a whole set of similar molecules. I'll now turn to some remarkable discoveries, of which Professor Wicksell has already said a few words before, that the variable regions of antibody molecules are themselves antigenic. The first one to show this, I think, was Henry Kunkel and his colleagues, who showed that myeloma proteins, which are antibodies produced by cancerous lymphocytes and which are monoclonal, that when you inject these myeloma antibodies into an animal, the animal will make antibodies against them, and these will react only with the myeloma protein of that particular patient and not with myeloma proteins made by any other person. And this work was extended by others, mainly by Jacques Houdin and his colleagues in Paris, who showed that ordinary antibody molecules made as a response to an antigen are immunogenic and will invoke the formation of specific antibodies if you inject them in another animal. Maybe it would help if I could have the next slide. You will see here I follow the traditional idea that there is a combining site which recognizes the antigen, but then as they showed that these variable regions are also antigenic, and not only that, but that there are a variety of different antigens in the variable region. I've just indicated them by these red spots. They're called idiotopes, and the whole set of them is called the idiotype of the molecule. That these are antigenic areas which, when injected into an animal, will evoke the production of antibodies against this molecule. And in fact, it goes even further. The animal itself, which makes antibodies to an antigen, will then start making antibodies against its own antibodies. But these antibodies, of course, also have an idiotypic profile. So the animal will continue to make antibodies against its own antibodies, and so forth. So we came to the situation where we have had to agree that the variable region of a single antibody molecule represents a whole set of antigenic determinants called idiotopes and a combining site by which the molecule recognizes another antigenic determinant or epitope. But we were getting into trouble when trying to interpret our experimental results. Let's have a look at the next slide, please. It's the most primitive picture I can make of a B cell. Here's a B cell. It advertises the antibody it can make. It's resting B cells. These receptors are antibody molecules stuck into the surface, and they're all identical. And I'll enlarge a little area of the surface of the cell in the next slide. There's one of these receptors. There's an antigen, some protein or so, which is trying to stimulate the B cell. And it's being recognized, part of it is being recognized by the receptor through this combining site that we still uphold. But the receptor itself has these idiotopic antigenic determinants in its variable region. This is the basic picture of the selective theories of antibody formation, and as most clearly formulated by Burnett. The antigen, we say, selects the cells which happen to recognize part of the antigen, and then stimulates these cells to produce the only thing they can produce, namely that particular antibody that is similar to the receptor. Of course, T cells are also involved. other factors, as I said, growth factors and so forth, but this is sort of the basic situation. Maybe we could have a look at the next slide. Everything is, I've just enlarged a little bit. Now I have here an antibody molecule which has been secreted by a B cell. It floats around in our blood and it recognizes part of this antigen with its combining site. And the antibody molecule is itself antigenic through its idiotopes. And it has a combining site, and it recognizes what I've called the epitope, an epitope on the antigenic molecule, some protein or hormone or some other molecule. Here may be another epitope, which may be recognized by some other cell. And I call this antibody 1. And I now use antibody 1 as an antigen to stimulate antibody formation against antibody 1. Next slide, please. And I then am confronted with two possibilities, which I've called alpha and beta. This is this antibody 1. And now there is a B cell that has a combining site which recognizes an idiotope of antibody 1 and will make an antibody 2, which is anti-idiotypic towards antibody 1. But it's also possible that this antibody 1, which is freely circulating, will recognize an idiotope on the receptor of a B cell. But since this antibody 1 was already recognizing the antigen that provoked it, and there must be some similarity between this idiotope and the original antigen. That is the beta situation. And it has turned out that they both occur. For example, if the original antigen is insulin, and you make anti-insulin antibodies, which is antibody 1, let's say, then in the beta situation, since this antibody 1 can recognize insulin, it now recognizes something on antibody 2, stimulates this B cell. It recognizes something that looks like insulin, and it has been shown that these antibody 2, which are produced, even function like insulin. And similar results have been shown for other hormones and other systems by others, Cazenave, Roland, Strasberg, Urbain, and others. Now, the point which I wish to make is, are these two situations really different? Is there a difference on this level of protein molecules between saying that antibody 1 recognizes antibody 2 or that antibody 2 recognizes antibody 1? Is there, at the three-dimensional molecular level, a distinction between recognizing and being recognized? So I have denied that and have said it's meaningless to distinguish that. And I conclude that idiotopes, these red spots, and combining sites are equivalent. And that an antibody has a whole set of idiotopes, which include what we traditionally called a combining site. In other words, an antibody molecule is multi-specific, has multi-combining sites. I don't have to belabor this point, I think, because it has been made repeatedly. What I should like to do is to introduce some numerology into the discussion. How large is the number of different antibody molecules that the immune system of a single animal or man can make? A mouse. This number during the past few decades has been estimated on more or less slender evidence, perhaps, to exceed 10 million. And this enormous diversity of different antibody molecules that the system can make is called its repertoire, the repertoire of the B cells. This repertoire was called by Coutinho complete, and by completeness he means that this repertoire is sufficient to recognize any molecule in the world, even molecules that the system has never encountered, as I said. Immunologists, they use terms which they have borrowed from linguistics, such as immune response, the system response. Now, if we look at languages, we find that the vocabulary of words in a language is rarely more than 100,000. In English, Shakespeare, for instance, who used the largest vocabulary in English, used about 25,000 words. So the vocabulary of words in a language is at least 100-fold smaller than the repertoire of the immune system. But now, if we consider that the variable region of an antibody molecule is made up of these two polypeptides, each 100 amino acid residues long, and that its three-dimensional structure folds into a set of perhaps 10 combining sites or more, it may be more reasonable to make an analogy between language and the immune system by regarding the variable region of an antibody molecule not as a word, but as a sentence. The repertoire of the B cells then doesn't become a vocabulary, but a lexicon of sentences, which is capable to respond to any protein, let's say, any other sentence that the system encounters. And at this point, I would like to make a few quotations from perhaps the most famous linguist at our present time, Noam Chomsky, who writes, and I quote, "The central fact to which any significant linguistic theory must address itself is this: a mature speaker can produce a new sentence of his language on the appropriate occasion, and other speakers can understand it immediately, though it's equally new to them. Grammar is a device that specifies the infinite set of well-formed sentences and assigns to each of these one or more structural descriptions. Perhaps we should call such a device a generative grammar, which should ideally contain a central syntactic component, a phonological component, and a semantic component." That's the end of this quotation. For the size of the set of possible sentences in a language, Chomsky uses the word open-endedness. And I now think that open-ended is perhaps the best description of the completeness also of the immune system. As for the components of a generative grammar that Chomsky mentions, we could, with some imaginations, equate these with various features of protein structure. Every amino acid sequence is a polypeptide, but not every sequence will produce a well-folded, stable protein molecule with acceptable shape, hydrophobicity, electrostatics, and so forth. There must be some, though not known to me, grammatical rules required. It's harder to find an analogy to semantics. Does the immune system distinguish between meaningful and meaningless antigens? It would seem at first sight that the immune system makes a response to a sentence presented by an invading protein molecule merely by selecting from amongst its enormous preformed antibody repertoire a suitable mirror image of this antigenic sentence. As you will know, Leonardo da Vinci, he wrote his private journal in mirror script with his left hand. Not every Nobel laureate is a Leonardo da Vinci, and it's in fact quite difficult without considerable practice to write and read mirror handscripts. I'll show you an example on the next slide. You see it says Stockholm, and then it says Stockholm in mirror script, and I now wish to impart upon you that these two would recognize each other, but since I can't write mirror script, I have helped myself out by just writing Stockholm once more with a red covering, and meaning that that's the mirror. That's also the principle of the following slides. The next one, please. You see here I suggest there's an antigen, which is a protein that has a certain amino acids sequence. Reading alcohol misuse can cause drunkenness. And now this antigen is going to stimulate the immune system, and it finds a cell that makes antibody 1, and the idiotopes of that receptor form a sentence of combining sites, which read, in January, Stockholm is usually quite cold. And you will see that here is a site where the two molecules recognize each other. And now I use this antibody 1 as an antigen and stimulate another animal. And there, this antigen now, antibody 1, finds a cell that has receptors with a set of idiotopes that reads every stockholder likes large dividends. And you will see that there is an area where the antigenic determinant is recognized by the antibody in the mirror script. Of course, this antibody 2 has nothing to do anymore with the antigen. So on the next slide, I'll make it all a little more complex. And there we use, as has actually been done experimentally, there's the antigen is insulin. It's a protein which has a sequence of amino acids, and it reads the production of insulin depends
on cells in the pancreas. And now this insulin is injected into an animal, which makes an antibody to insulin. And this antibody happens to read the name of Insulinde, was given to Indonesia last century. And you will see that there is a combination possible. So that would then be the combining site in traditional terms of the antibody. But now we make an antibody 2. We use antibody 1 as the antigen, as before. And here, for instance, here's a cell that can make an antibody 2 alpha, I call it. This case of amnesia lasted quite a long time. And here is a recognition between the two. But of course, this one here, this antibody 2 alpha, has nothing to do with insulin. But there's another one which reads diabetes can be a result of insulin deficiency, and this combining site, which recognizes antibody 1, is the same as this part of insulin itself. So this would then, in these experiments, be an antibody, an anti-antibody, which functions like insulin.
I should therefore... No, there's one thing I'd like to emphasize.
It has been estimated that
A human individual produces about 10,000 different proteins, namely enzymes, hormones, cell surface proteins, and so forth. But at the same time, we estimate that the immune system maintains a repertoire exceeding 10 million different proteins, namely the antibody molecules. Therefore, the antibody molecules as proteins are 1,000-fold more than all other body proteins taken together. I should therefore like to conclude that in its dynamic state, our immune system is mainly self-centered, generating anti-idiotypic antibodies to its own antibodies, and because they constitute the overwhelming majority of the different proteins in the body. The inheritable deep structure of the immune system is now known. We know that certain chromosomes of all vertebrate animals contain so-called V genes, which code for the variable region of the antibody molecule. You can take this slide off, please. And there are other DNA segments which participate in encoding the variable region of an antibody molecule. Furthermore, experiments in recent years have demonstrated the generative capacities of this innate system that's encoded in our genes. In proliferating B lymphocytes, these DNA segments that code for the variable region are targets for somatic mutation, which result in the formation of antibody variable regions which differ in amino acid sequences from those encoded in the stem cell from which these lymphocytes have arisen. Maybe I could have the first or last slide, which you haven't. No, the next one. You have already seen this. What I was trying to impart is that the resting B cell is encoded by genes
in our chromosomes, in the chromosome of the zygote. But as the B cell proliferates into perhaps a large clone, these genes are subject to somatic mutation, which enlarges, of course, enormously the variability that can be expressed. And experiments have shown that if you look at the gene of one of these cells that is producing antibody, you often find that it differs from it differs in 5 or 10 places from the gene that was present in the chromosomes of the zygote. But it's still possible to identify that gene. And that we might call the etymology of the immune system. You can find back to the origin. When we place a population of lymphocytes from an animal in tissue culture fluid, and then we add antigen, the lymphocytes, if the conditions are right, will start producing antibodies against that antigen. In the absence of any nerve cells, I find it astonishing that the immune system embodies a degree of complexity which suggests some perhaps superficial but striking analogies with human language, and that this cognitive system of that enormous complexity has arisen without assistance of the brain. It seems a miracle that young children can learn the language of any environment into which they are born. The generative approach to grammar pioneered by Chomsky argues that this is explicable only if certain deep universal features of this competence are innate, he calls it innate characteristics of the brain. Well, biologically speaking, this hypothesis of inheritable capability to learn any language means that it must be somehow encoded in the DNA of our chromosomes, as is the immune system. And should Chomsky's hypothesis one day be verified, then linguistics would become a branch of biology, and perhaps someday in the far future, the humanities perhaps would become part of the natural sciences. Thank you.